So good afternoon, everybody. I hope you are all doing well.
Good afternoon, ma.
Yes, God bless you. And I hope you are coping well with your series of lectures. I pray that God will see you through. So we'll just quickly go through the remaining lecture, which shouldn't take us more than 15 minutes. But I want to implore you to please try and read this part thoroughly, get familiar with it, and have it at the tip of your fingers so that by the time the next lecturer comes in, you are already used to this lecture notes. It is not nice to pile up your notes so that when exam comes, you will not be having problems. So I want you all to please go on silence because some people have started again. Please silence your phone so that we can have a smooth lecture. I'll go ahead and share my screen.
Sorry, man, you can mute everyone.
Ìpè kan ti lọ sọ́dọ̀ àwọn ọmọ orílẹ̀-èdè Nàìjíríà pé kí wọ́n máa sọ ìṣòro tó bá ń kojú wọn síta dípò kí wọ́n máa gbé sínú èyí tó ń ṣe púpọ̀ ọ̀pọ̀lọpọ̀ ẹlòmíì. Bákan náà ni wọ́n ti rọ àwọn ọmọ orílẹ̀-èdè Nàìjíríà pé kí wọ́n máa ṣe ìrànlọ́wọ́ fún ẹni tó bá ní ìṣòro nítorí pé eléyìí yóò ṣe àdínkù sí ìtẹ̀lé ara ẹni tó ń ṣẹlẹ̀ láwùjọ wa. Èyí ni ọ̀rọ̀ àwọn tó ti àtẹ̀jíṣẹ́ ránṣẹ́ nínú kókó tó ìwé ìròyìn abala àgbéyẹ̀wò wọn ní nítòótọ́.
All right, thank you very much. So I guess we can start our lectures right now, and then I will release you in the next 15 minutes, hopefully. So last week, remember, we started on Mendel's law. We started on Mendel's law, and which I muted everybody. Some people have unmuted themselves again.
It's not mine. Hey.
All right, so last week, quickly, we started on Mendel's law. And first of all, we look at all the characters that Mendel has studied. He studied dominant traits, which include the purple color of flower. For example, he also studied the seed color, which could be yellow, the the seed shape, which could be round, and the pod, which could be inflated, and he also studied the flower position, which could be axial. And we noted that, for example, the purple color is dominant over the recessive white color, and the yellow color is dominant over the green color. The green color is recessive. The round seed shape is dominant over the wrinkled shape. Then we went ahead to discuss the meaning of or the representation of these various characteristics with letters. That Mendel uses pairs of letters to designate the genotype of the character that he's studying. So from your knowledge in secondary school, you would have noted that they usually use capital letters for the dominant colors and small letters for recessive colors. So if we are talking in terms of tallness, for example, capital T is for tallness, small t is for short. And if you are talking in terms of yellow, capital Y is yellow color, while small y is for green color. So if you have one T, one capital T and small t, that means the person is dominantly tall, but has some short recessive gene. And if you have capital Y and capital Y, that means that that seed is completely yellow in color. There's no recessive gene with this double capital. If you have two small s, for example, that means that that flower is entirely short. There's no gene for tallness. So, and we also looked at that any allele that have double capital T is completely tall, double small t is short. So when you have the same letter, for example, TT, capital TT, no gene or allele for shortness, it means it's an homozygous allele. So two capital T is homologous, two small t is homologous because they are clean. There's no mixture of tall or short. But when you have capital T and small t, it is heterozygous because they are hybrid. They combine both tall and short gene. So that was where we stopped last week. So we want to look at the first law of Mendel, which is the law of dominance. Law of dominance. And what does the law of dominance state? What does the law of dominance state? The law of dominance states that in a heterozygous condition, that means when you have one dominant and one recessive, that the allele whose character are expressed over the other allele is called the dominant allele. I repeat, the law of dominance states that in a heterozygous condition, the allele whose characters are expressed over the other allele is called the dominant allele. For example, look at this flower. This is a parent generation. The parent generation of this flower has red, that is dominantly red, and the recessive white. So one of the partner is red, completely red, and the other one is completely white. I don't know why I unmuted everybody and some people are unmuting themselves and disturbing the class. All right, so from this generation, all right, from this generation, the complete red and the complete white are coming together to donate gene to have a generation, their offspring. So the complete red donates an R, that is capital R, responsible for red coloration. And the white donates a recessive white, small r, responsible for white color. But from his study, the generation that develops from there are red in appearance. They are red in appearance, meaning that that red color supersedes or is dominant over the white color. Just like last week, I gave you an example that a dark in complexion man may marry a white in complexion woman, and the child can be light or black in complexion. For example, if they have 4 children, one of the children may be completely dark in complexion. 2 of them may be completely white in complexion, and one of them may be just mixture. I mean, neither completely white, neither completely dark. And you may have 3 very dark and 1 very light. Okay, so in this case, the generation, which is the offspring, appear red because red color dominates the white color here. But if you look at this, you will discover that despite the fact that this flower appear red, it has a recessive gene for white. So this gene, even though it is recessive, it is not showing in this flower, but it has a white gene, which could come to show later in generation 2 of this flower. So this is what the law of dominance is talking about in heredity. All right. So now we have, I want to explain the Punnett square, the Punnett square, Punnett square. It is also simply called P-square, P-square. It is named after the founder, Reginald Punnett. And the Punnett square is a square diagram that is applied for the prediction of the genotypes of a particular cross or breeding experiment. The square diagram is employed by biologists to determine the probability of an offspring having a particular genotype, like the one I showed you earlier. The parents, tall and short. You can see one parent is completely tall, the other one is completely short. Where T is the dominant allele, like I showed in that flower, for tall stems, and T is recessive allele for short stem. The P-square for this cross, for example, if we have TT and T small, I mean 2 small t, the other one we use is red coloration. Is red coloration for the flower. But we are looking now at another one. It's still the same thing because capital T, small t. So look at it, try to cross it yourself as you are looking at it. Look at the parents, TT. These are the alleles for the parents, TT. And then the short alleles, I mean, for the short parent, these are the alleles, small t and small t. So this is how the crossing is done. T, 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 T. Then capital T and small t, capital T and small t. So you will notice that in this crossbreeding, all the generations arising from these parents are looking tall. They appear tall. All the 4 of them appear tall because they all have both the dominant gene and the recessive gene. But if you look at these 4 closely, each of them have a recessive gene for shortness. So we can say that 100% they are dominantly tall. Okay, that is how to explain the P-square. But of course, we have done this and we have seen that this generation appears red, but has some recessive gene in it. Okay, so we'll go ahead for the second law. The second law is law of segregation. You will discover that when I showed you this, we did not segregate. We did not segregate. But from experience, we know that only one R will be donated here, one R will be donated by the recessive, and you have it. But the P-square have shown us how the gene separates. I've told you that under where I showed you chromosomes and genes, and you have seen the double helix, all right? So now, looking at the law of segregation, this is TT, that is from that our first generation. Remembered that we have offspring that appears tall with dominant tall trait, but with small recessive tallness. So we want to separate those two now in the law of segregation. Look at the T comes here, the small t for shortness come here. And so that is if in the second generation, the two heterozygous alleles are coming together as parents. So one parent here, one parent here. Separate the alleles, T capital, T small. Here too, separate it, T capital, T small. So now cross both. So what will happen? T, you have two capital T's. Then capital T and small t, you have capital T and small t in this box, in the Punnett box. Then pick the small t, this small t, cross it. So you have capital T and small t. Then this small t is crossing this. Then you have two small t. So in this Punnett square, what does the second generation offspring look like? You have one of the offspring that is completely tall, dominantly tall, because the two alleles are tall. There's no recessive gene in this first one. In the second one, you have heterozygous. So this is homozygously tall. This here, you have heterozygous, where you have both the tall and the short offspring. The same thing with the third one, heterozygous. You have alleles for tallness and shortness. But for the third one, it is a completely short offspring because both the alleles are short. So this is predominantly a short offspring. So in this case, you can say, how many tall offspring does these parents have? They have 3 tall offspring and 1 short offspring. But in the tall offspring, what happened? You have 2 of them that have alleles for shortness. They have this gene for shortness. So you can say that completely tall homozygous is 25%. Then the heterozygous tall is 50%, 2 boxes, while the homozygous short offspring is 25%. So, but for the total offspring that are tall, they are 75%, 1, 2, 3, 3 boxes. So that is how this is explained for recessive phenotype, that is outward appearance, and for tallness and shortness. So, and we go to the third law of Mendel, which is the law of independent assortment. Law of independent assortment. Here, the law states that inheritance of one pair of factors, that is the gene, is independent of the inheritance of the other pair. Alleles for different traits are distributed to sex cells and their offspring, independently of one another, as we have seen in what we have done. So the law of independent assortment really emphasizes that there are separate genes for separate traits and characters, and that these genes sort themselves independently of each other. That at the time of gametes and zygote formation, the genes are independently passed on from parents to their offspring. And so during his work, Mendel discovered that the height of the plants, the shape of the seed, and the color of the pod does not impact on each other. That is, the fact that you are tall will not determine your shortness. That is, the gene for tallness and the gene for shortness, they are different from each other and do not affect each other in any way. That is what it is saying. That is what it is saying. I don't know whose computer is that.
Sorry, man, you can mute everyone so that they'll be able to unmute themselves.
I'm doing it. I'm even not allowing the person to unmute, and the person is still muted. I mean, the person is still on mute. I hope the person will not unmute again. All right, so that the gene responsible for one character actually differ from each other, and they do not affect themselves in any way. So the fact that you are short does not mean that you must be dark in complexion. Those two genes different from each other. Jesu Numi Sarah Adebisi, well done, oh. Well done. I will advise you to cover yourself because you are virtually naked. Or please, remove your video. All right. So the fact that a plant is tall does not indicate that the plant must have a green pod. Neither must the green pods be filled with wrinkled seeds. So each of these genes differ from each other, as you can see. Dada Olanike, type your question. I'm not ready to unmute anybody for now. Disturb this class too much. So looking at this law of independent reassortment, that takes us to our last explanation, this one. So you can have different genetic combination. Like we have said, one gene, the gene responsible for one character does not affect the gene responsible for another character. Each gene will play out independently. So look at this parent generation. Okay, first, the law of independent assortment states that in a question involving two different traits, your answer will be independent assortment 9 times out of 10. You first of all find out this is what the law states that I've stated earlier. But what happens with the law of independence is that in a question involving two different traits, like look at this one now. It's talking about maybe the color, or what did we see? Let's look at it, let's confirm so that we'll not do the wrong thing. Okay, so the seed color may be yellow or green, recessive. Then it may be round or wrinkle. So that means the seed color yellow, and then recessive will be small yellow. And for round will be big R. For wrinkle will be small r. So let's go back to that table. So look at it, yellow color, then round. So for this seed, it has the gene for yellow color, dominant yellow color, and dominant round seed. For the other parents, it is recessive yellow color, that is, it is green. And then it is wrinkled, as you can see, with small r. So the small y is representing green color. The small r is representing wrinkle shape. So when they want to come together to give birth to their first generation, what will you have? You have a color that is yellow and a seed that the color is yellow and it is also round. As you can see, it will donate one of these genes, color Y. And then the small one, then the big round and the small round. So in the first filial generation, the first offspring, the seed appears round and it appears yellow. But if you look at it closely, you will see that even though this seed appears yellow physically, phenotypically, and it appears round phenotypically, it also has recessive green color and recessive wrinkles. Even though the phenotype, the physical appearance is yellow and round. So the next generation, if you have this now and is getting married, let's assume, you know, this one is a plant. Let's assume that this first generation, the child that this generation gave birth to, that now have both that appears, no, heterozygous for yellow, but with green and wrinkle color. Let's say two of them are now getting married or coming together, or they want to crossbreed them. Let's now segregate their gene because they appeared yellow and round. But we already said that they have some recessive gene. So this is what their gametes will look like. The gametes that will come together will be heterozygous yellow, heterozygous round, then heterozygous green, and homozygous round. You are separating these genes. And you know how you do it? Pick this and pick this. Pick this small and the big R. Then the big Y and the small r. To separate the gametes, the gene. And of course, you have it on this side likewise. Then you begin to cross. This is the gamete from heterozygous parents, another gamete from heterozygous parents. All these are included in the filial generation, but now you are having two of them. So one is here, one is here. Then you practice crossing on your own. So you can see that this first one, you have capital Y, then capital R. So this organism is completely yellow and completely round. There's no recessive gene in this offspring. For this offspring, you have, it looks yellow and round completely from this cross, when you cross these two together. But it has a bit of green color because of this small wire that is showing up here when you cross this. When you cross this with this, you have a completely yellow color and a green color plant that have a recessiveness for wrinkleness. The same thing here. You have a seed that is yellow in color with recessive green, round with recessive wrinkleness. So by the time you get here, you have a yellow seed with a recessive green color, completely round. Here, it is completely green because all the yellow colors are recessive. And then you have homozygous round, wrinkleness. So they are all wrinkled. So by the time you do all this cross, you will discover that you will have phenotypically, that is physical appearance. You will have 9 yellow seed, which is completely yellow. You will have 3 completely green seed. You will have 3 yellow but wrinkled seed. Then you will have 1 completely green and completely wrinkled seed. If you go home, I have sent your notes to you since yesterday. So if you collect your notes, just assume that you are not seeing the answer here. Close your eyes to this cross that has been provided for you. You just make do on another sheet of paper with your parent, with the parents, this and this that has been provided for you. So you try crossbreeding them yourself with this and this, and try to see if you will get the cross completely. But it will be nice that you will start practicing with Punnett square. So looking at the Punnett square, don't look at the answer you have. You just prepare your Punnett square like 4 square. You may even ignore this TT. You may use your own, you may use another capital letter. Maybe it's BB you want to use or whatever, and use it to practice and try to cross it. So for those of you that may likely have, though we are dealing with microorganisms, is the same type, it is the same thing that plays out, whether you are talking about plants, whether you are talking about animal, whether you are talking about microorganism. It's the same thing. It's genetically, it plays out this way. So you can actually check for a person that an AA that is marrying an SS. What is the probability of having an SS child? You can play around with colors. You can play around with different traits, and then you can apply it to microorganisms. So the way the botanists or agricultural people are crossbreeding plants, the same way microorganism crossbreeds. For example, if I have an organism, like I have one really, it is an actinomycetes, it is a streptomyces. It has the gene to perform different metabolic activities that are positive, but it is a slow grower. It does not, sometimes it doesn't grow until after 5 to 14 days. And it has this good metabolic, the gene for breaking down some of these toxic substances in the environment. So supposing we have the equipment, what am I supposed to do? I'm supposed to look for another bacteria that is a fast grower, probably that grows within 12 hours or 18 hours or at most 24 hours. That organism grows fast, but does not have the gene to perform the role I want. So I could take the gene from the actinomycete and insert it into the fast grower so that I will be able to generate this organism, this recombinant organism to give me the desired characteristics that I want. And that is what microbial engineering or microbial engineering, genetic engineering in microbiology is all about. The same way crossbreeding takes place in plants and in other animals. The same way you can apply it to your microorganisms. So some of you might want to grow up, whether you are in botany or you are in zoology or you are in cell biology. Of course, cell biology, they know they cut across. Microbiology also cut across because you have microorganisms in plants, in humans, and in other animals. You may want to go into genetic engineering. The botanists or the agriculturalists have already gone ahead because most of these food items that we have are as a result of genetic manipulation or genetic engineering. When we were growing up, when we were very small, the only color of maize that we have was white maize. But along the line, by the time I finished my secondary school, we suddenly started to see yellow corn, yellow maize. It was as a result of crossbreeding. Then later, I began to see purple maize, even though it is not common, but it is there. The same thing, we used to have all the pineapples short but sweet. But now they have crossbreed by introducing the sweet color into the tall pineapple. Now you have pineapples or oranges that could be very big and sweet. You can crossbreed. The same thing we had beans. We used to know of that white beans with black face. Over time, we started seeing Ife brown. After Ife brown, we started to see Oloyin, different types of names that I don't even know their scientific name now. So the agricultural people have gone a long way. The same thing, they are crossbreeding chicken, crossbreeding different animals together. And microorganisms, the same thing, especially in the aspect of removing the metabolic genetic character that is of benefit from one organism and introducing it into another organism to achieve your purpose, like the growth rate I told you about. So you can actually manipulate this organism, and that is the essence of genetic engineering and all this heredity and gene and so on and so forth that we are studying. And that brings us to the last lecture on my path. So even if I will see you again, it will be maybe for one reason or the other. Probably one lecturer could not come and I'm asked to assist or not. That could be the only reason why I will have to take you again. So for that, definitely in January, another lecturer will start the lectures with you. But please don't miss your practicals, because your practicals form your continuous assessment. And it will be nice for you to attend your practicals. I have also given your notes to your class captain. Ensure that you key into it and take all your notes, and do not abandon your notes. Try to read your note ahead of time so that by the time your exam starts, you will not be too much in a kind of mess. All right, so I will just stop here and let me now allow you, if anybody wants to ask any question, I will allow you to unmute. Okay, if there's no question, that brings us to the end of the class.
Thank you, ma.
Thank you very much. I wish you a Merry Christmas, a nice holiday. Please be careful and stay pleased.
Thank you so much, ma.
Bye-bye. God bless you.
Bye.
Bye-bye, ma.
Bye-bye.
Thank you, ma. Thank you.
Thank God.
Ma. God bless you, ma.
Amen, and you too.
Thank you, ma.
Thank you. Bye-bye. God bless you.